Okay, good. Good morning, everyone. So today is the last day of Gyan course, particularly for this course. Basically, there are another course is going on in MNIT as well, organized by Department of Computer Science, probably. So Dr. Bipro has completed his syllabus or topic for this Gyan course, and he will take another maybe half an hour just to recollect all the lectures and clearing doubts and all those things. But of course, for today's exam, majority of the question is like 90% of the questions will come from Bipro's lecture. Okay,
so in the test there could be some numerical or just about the concept?
Maybe, but it's mostly with the concept. It's like I think 90-95% of MCQ is on concept only.
MCQ
Best.
or match out the following?
Yes. So anyway, so I prepared a different slide at the beginning. As per the time schedule, we had a talk I think on first day, but whatever was added, Bipro almost covered everything. So last night I had to change the whole slide, and so today I will cover mostly about bioremediations and phytoremediations, the very basics for faculties. I mean, they probably knows everything if they are from biotech background. Maybe for environmental engineer there will be still something new. So for bioremediations, we know that basically whenever we are talking about wastewater management or wastewater treatment, there are chemical method, there are physical method, there are biological method. These are the three main methods we follow basically. So bio means to remediate any pollutions or to manage waste, different kind of waste. It could be air, it could be water, it could be soil or solids. Okay, we use some biological organisms. It could be bacteria, it could be fungi, it could be microalgae, it could be plant, anything. And so when you are talking about bioremediation, that means we are covering all parts or all different form of living organisms. Now if you want to be very specific, there could be like if you are using bacteria for bioremediations, we can call it microbial remediations. If you are using algae, or then we can call it phytoremediations. All are bioremediations under the one umbrella, but depending on what you are using, basically what working force you are using, whether it's a bacteria or fungi. Like for plant, you can call it bioremediation, there is no wrong in it, but if you use phytoremediations, that is more appropriate for that particular term. Okay, so for bioremediation there are different types. One is in situ bioremediations and another one is ex situ. So from the name itself you can understand in situ means it will happen in the same locations. Like if this garden just outside your EDC, if this got contaminated, sudden spillage of chemicals, toxic chemicals, so we'll do the treatment of this garden here itself. While in ex situ is we carry the toxic material or water to a different location, like basic biological wastewater treatment. It's not like we are treating the wastewater at the same locations. Every wastewater goes into a STP plant, so it's a different location, isn't it? So that's why there is a two different process, in situ and ex situ. In situ we basically use phytoremediations for like this garden. If this got contaminated, you can use, you can add some plant in this garden, specific plant which may uptake the contaminant through its root, which can immobilize that contaminant, or we can add some certain bacteria, microbes in the garden which can degrade or immobilize the plant materials. Okay, so for in situ bioremediation there are two things. One is like intrinsic bioremediations, okay, and another one is extrinsic. So in intrinsic we basically it's like microbes. So it's like whatever microbe is present, they are more than sufficient to do the treatment. Okay, so we don't need to add anything. So it's basically one of the means the best possible scenario, but only thing is to enhance the degradation or the bioabsorption rate, we can change the environmental conditions, like we can increase the moisture, we can add some carbon source into the garden. It's like to stimulate the microorganisms basically, so which is nothing but the known as biostimulations. Okay, it's like to provide the best possible environmental conditions, nutritional conditions to the microbes, so that you have the optimum rate of remediations. So then you have of course the engineered in situ bioremediations. So engineered means you did the survey of this particular garden and you find out that the microbes which are present, they will not survive in the toxic conditions. Okay, so in that case, what you required is you need to add microbe from outside. So it could be a indigenous microbe, or like in a, what is happening nowadays, so many people are doing genetic engineering. So you can specifically design some microbes and you can add into the sediment or soil to remediate it. But one thing is, when you are doing this addition of microbes, it has to be always combining with biostimulations. Okay, because you are adding some specific microorganisms, so that means they also need some specific condition for it, right? So in engineered in situ bioremediations, you always have to combine bioaugmentations and biostimulation to provide the best possible atmosphere for the added microorganisms. Okay, so this is a basic chart or basic process we follow. So we first, of course, as soon as it get contaminated with maybe it could be chromium or it could be some pesticides, whatever it is, we first always has to go with the survey, microbial community analysis, to find out what kind of microbe present. You can do a basic toxicity state, whether those microorganisms will be able to survive or not, and then based on that you will decide what kind of microbe you need to add in a small scale lab and ultimately apply into the large scale procedures. Okay, and then the ex situ bioremediation, of course, this is the mostly with the biological wastewater treatment. Okay, so you have to carry the things, but the main problem with ex situ is like solid waste management. Of course, we are not treating the waste just next to our houses, municipality person coming and carrying all the waste into different locations and treating it. But the issue, one of the major issue is, particularly in third world countries or developed countries, is when they are carrying it, they don't carry it properly, isn't it? It's like half of the garbage remain on the streets basically. It's happened in MNIT as well. Okay, so that is one of the biggest issue. So that's why it's like, suppose you are dealing with radioactive waste, so carrying the radioactive waste is a very, very serious challenge. If you have any leakage or accident or spillage, then it can cause a serious damage to the environment or the people as well. So then that's why for radioactive waste we always prefer in situ remediation kind of technology. Okay, like in those who have this nuclear disaster, it's like they always use some microorganisms or fungus to treat, and then people are also extracting or isolating microorganism from those areas as well, because after maybe 2-3 years the microbe become very, very resistant against those nuclear waste. So you can use those kind of microbe in some other place as well to treat the radioactive waste. Okay, so for bioremediations, this is the basic mechanisms. So whether it's organic or inorganic, like metals, so the first thing comes is bioabsorptions, followed by bioaccumulations, bioprecipitations, as well as transportations or biodegradations things. Okay, so the bioabsorptions, you can see it's happened on the surface, so it's a surface phenomena basically. So for bioabsorption, it doesn't mean, or you don't need to have living organisms, you can go with the dead biomass as well. Okay, so it's like bioabsorption, so you have the basic absorptions, and bioabsorptions means instead of using any chemical or other kind of adsorbent, we are using all biomass, biological absorbent. Okay, it could be dead bacteria, it could be dead plant biomass, It could be biochar is a very good example of bioabsorption process. So you can see that there could be a different mechanism for bioabsorptions like precipitations of metals, ion exchange, complexations, all different kind of mechanisms are there. Of course, once you do one experiment to remove the metals, and then you can do other experiment to find out that what mechanisms is responsible for the bioabsorption process, what kinetics it is following. For bioaccumulations, so in bioaccumulation what happens is microbe uptake the contaminant inside them. So uptake means the microbe has to be live or living biomass, okay, because the metabolic activity has to be going on, okay, then only it can uptake the metal or organic contaminant inside them. So of course it's not like that, suppose you are working with chromium, so it's not like that microbe are resistant to chromium, they are toxic, but they, depending on the what kind of microbe is you are using, their toxicity may vary. One microbe may be toxic to 1 milligram per liter chromium, one microbe can survive even at 50 milligram per liter of chromium, okay. Or if you are working with solid, it will be of course be milligram per gram, the unit will change, okay. So that means the microbe will have or has certain detoxification mechanism when they are uptaking it, isn't it? So when they are uptaking it, it could be like for metal, they can reduce the metal to a lesser toxic form, like chromium. Chromium 6 is toxic, but when you reduce the chromium to chromium 3, it's become less toxic. It's not like that chromium 3 is non-toxic, but it become lesser toxic, okay. So you are reducing their impact on the environment, okay. Similarly for arsenic, arsenic 3 is extremely toxic, but if you oxidize it to arsenic 5, it become less toxic, okay. If you go with like other metal like selenium, sulfur, mercury.
Sir, can these all three procedures like bioabsorption, accumulation and transformation be happen on the same cell?
Yes, so if you are working with living biomass, so generally bioaccumulations is followed by bioabsorption. It's not like that microbe will directly go bioaccumulations. It will bioabsorb first on the surface of the microorganism, then they will uptake. So the bioaccumulation. Then you will have the biotransformation or biodegradations. Okay, so that is actually bacteria or fungus, whatever you are using, their detoxification mechanism. Okay.
Transformation actually happen on the outer side, or it can be?
It could be internal, it could be external as well. Like for reduction of metal, it could be on the surface, okay, or it could be internal as well. Okay, one of the example I can give you like selenium. Okay, so like Dr. Ashutosh is from Punjab, and he knows that in selenium is a huge problem in Punjab, okay. So for the selenium treatment, what we do is selenium, of course you have metal has a different oxyanions, so selenium 6 and selenium 4 is extremely toxic, okay. So for microorganism, for their bio detoxification mechanism, what they do is they reduce the selenium to its elemental form, okay, that is selenium 0. Now for selenium, it is easy to understand their mechanism, whether it is happening internal or external, because when selenium is reducing from oxyanion to elemental selenium, it forms nanoparticles, okay. And if it is external nanoparticles, if the reduction is happening externally, the nanoparticles is outside, okay. It's very easy to collect the nanoparticles.
Is there any particular component, compound or element that has role in the reduction of chromium 6 to something?
Different enzymes are present
In case of living?
in case of living. Of course, in the reductions you cannot go with the dead biomass. For dead biomass, it's only the bioabsorptions, because all other bioaccumulations or biotransformation, you need a living microorganisms where microbes is alive, they are doing their own metabolic activity, okay. Otherwise you don't have the accumulations, okay. In some cases in bioabsorptions, if you have some other chemicals, then you will have a chemical reductions. Their microbes only role is to immobilize the metal, whether it's a reduce or not, okay. So there you will not have a bio reductions, that will be a chemical reductions, like in bioprecipitations. So So if you see, if sulfide is present, if metal is present, they will automatically form metal sulfide by chemical reactions and they will precipitate. The role of bio here is the bio immobilize it. In a simple precipitation, it will be difficult to remove the precipitate. Okay, it can easily leave. You cannot restrict them. But if you are using biological organisms, they can restrict or they can immobilize that precipitate on the surface of it. Okay, so it's become very easy to remove. The precipitate sometimes has become very difficult. You cannot separate. Okay, while in case of biological one, you can easily remove them. So your removal efficiency as well as the longevity become much higher. Not only that, in bioabsorption you also have desorptions. Okay, so desorptions mean based on if you change the pH or if some environmental conditions change, the metal can start leaching again. Okay, so that is much beneficial for bioabsorptions, because in bioabsorptions the desorption chance is much lesser compared to a chemical adsorbent. But in an experiment you also need to check desorption as well, that at what conditions or how stable your bioabsorptions is. Okay, how long or at what conditions, till what conditions. Suppose you are doing the bioabsorption at pH 4, so you have to test at different pH condition to study the desorption as well, because in environment pH can change based on the what kind of waste is depositing over there. Okay, so these are the, so as you see, like it's not just about living organisms, basically bacteria or microbes, they follow all different kind of mechanisms for the detoxification mechanism, otherwise bacteria won't survive as well. Okay, so for if it is the internal detoxifications, after reductions bacteria can accumulate up to certain point, but then they also release it. Okay, so like the one selenium what I was talking about, so if it is a external reduction by bacteria, the nanoparticles after reductions they are outside. And the good thing about selenium reduction is you don't need any UV visible or anything to test whether reduction is happening or not. Okay, as soon as selenium reduce into elemental selenium, it turns red nanoparticles. So color completely change from transparent to reddish media. So you will understand that whether the reduction is happening or not, and the color change slowly. It's not like completely red immediately. So the reductions, higher the reductions, the darker is the red. So from light red to the medium red and then complete dark red. Okay, that means it's like you have almost 100% of reduction of selenium. So when the external reduction is happening, it become very darker red, but when it is internal reductions, so bacteria is accumulating the selenium, then the red color is much lesser. Why? The selenium nanoparticles are inside, bacteria is accumulating it. But of course nanoparticle is also toxic to microorganisms, isn't it? So bacteria cannot accumulate as much as the one. After certain time what will happen is the cells erupt or disrupt, and then nanoparticles start coming out into the media. So you can see the time durations and the change of the difference in color also tells us about whether it is happening internally or externally. Okay, so these are some notes, those who wants to study later. So for bioabsorptions, basically in normal absorption as well, pH is very important. Okay, because based on pH you will have the media or the aqueous system you are using, you will have either higher proton or lower proton, isn't it? Based on that it will influence your adsorption as well. Okay, but not just about your aqueous media, one of the major factor for metal bioabsorption is metals speciations. So this is one example, like for lead, if suppose you are using lead bioabsorptions or lead adsorption in chemical. So if you see here, like above pH 7 or not exactly 7, it's like 6.4 or let's just say 6.5, the lead is, and above 0 is your aerobic conditions basically, and less than 0 is your anaerobic conditions. So whenever you are working in lab, generally we are working in flask, so it's aerobic conditions, right? So if you see, if pH is your above 5, if here you can see that lead is not present as a lead ion, isn't it? They will automatically precipitate if carbon is there in your media as a lead carbonate. Okay, so these precipitations is not happening due to adsorptions, isn't it? So this is happening due to complete chemical precipitations. It's like if you are you did one experiment at pH 5 and you see that you did maybe you did at experiment at pH 5 and you see that, well, you are using some biochar, your removal efficiency is 55%, and someone did a lead adsorptions with biochar with maybe at pH 8, and they said, well, I have 100% efficiency. But the thing is, that is a overestimation. Why? Because their lead is actually precipitated immediately, not a bioabsorption process. Okay, so that's why in bioabsorption pH is very important, and whatever metal, and best is for lead, this is your Pourbaix diagram. For cadmium it will be different, for chromium it will be different. So it is very, very important to know that at what pH your metal remain in its ionic form. Then only we will be able to say that whatever removal is happening, that is happening because of bioabsorptions or adsorptions, not because of precipitations.
You have the chromium diagram.
Yes, yes. I mean, you don't need any. Just go to Google image, search chromium Pourbaix diagram, and you will get it. For any metal, you will get this kind of things. But the thing is, not just the pH, but you also need to know that what you are adding in your media. In your media, if the carbon is not there, suppose you are using it plain water, so of course it will not precipitate, isn't it? But when you are working with real wastewater or a synthetic wastewater, you will have all those different carbon, bicarbonate, or all those different kind of carbonic form, and it can precipitate. Similarly, not only that, if you see, it can also have, of course, oxygen is there. Lead oxide precipitation can happen depending on the how much dissolved oxygen is present. So that's why, so we generally follow, like for lead, we always try to have the pH less than 6 when you are studying lead bioabsorptions. Okay, so Pourbaix diagram is very, very important for your bioabsorption study. So this is the basic difference or comparison with bioabsorption and bioaccumulations. So the first thing first, bioabsorptions happen with your dead biomass. So that's why it's a passive process. While for bioaccumulations, you have to have a living biomass, because you need microorganism to be metabolically active. Okay, otherwise there will be no accumulations. Second thing is, the another measure is where it is. It's a bioabsorption is like it act as a very similar to the chemical adsorption. So it is a very fast. So when you are doing like when you are starting with the bioaccumulations or bioremediation of certain metal, in our experiment, if we want to study the mechanism, we always have different kind of controls. Okay, it's like one control, like suppose you are studying with lead biomass. Okay, so one control will be the microorganism autoclaved killed. Okay, so everything is same, lead concentration, media, everything is same, but their biomass, the bacteria will be using, you need to kill it first by the autoclave, and then we add the same biomass into this. So whatever removal will happen lead, that is due to the bioabsorptions, not by accumulation or anything. Then we can add another one, same thing without biomass. Okay, so if some removal will happen, that means the removal is happening due to chemical precipitations. Okay, then you can have same living biomass but without carbon source. Okay, so that will tells you about bioabsorptions plus if some bioaccumulations happening or not, but it shouldn't happen because you are not adding any carbon source. So bacteria is alive, but for to run their metabolic activity, that will be extremely slow because they have no carbon source. Okay, so your treated examples, whatever removal is happening, it's happening because of bioabsorption, chemical precipitations, bioaccumulation, everything. So when you have this kind of control, then you will understand, you try to minus it, total removal this, bioabsorption removal, total removal maybe 80%, bioabsorption removal 10%, chemical precipitation 2%, then you will understand if you minus it that well the 70% is bioaccumulations, biotransformation, biodegradations, all those things. Okay.
What will be the calculation for the dead biomass?
Is the simple bioabsorptions. So that is your
If there is any kind of transformation happening.
no. So transformation in the sense reductions.
Yes.
So that will tells you in the chemical one if chemical is happening or not. Because in bioabsorptions whatever is happening is we are doing with the dead biomass. Okay, so that we calculate as a total removal for bioabsorptions. Okay, it's go with the bioabsorptions and bioaccumulations.
Actually, I wanted to ask you that if I am doing with dead biomass and I am doing with chromium 6, okay, so if there is some kind of reduction happening, so how can I say that that chromium 6 is reduced to chromium 3? How much percentage? 30%? 40%?
That you can have to go simple chromium analysis. You have different UV visible method where you can measure with adding of different chemicals. You can measure how much chromium 6 is there, how much chromium 3 is there at the end. And then in the biomass, if you see like if it is happening, you can go with XRD or you can go with XPS to understand what if some is the chromium, what is the speciation of chromium in the biomass. In the liquid media, you can simply go with the UV visible method. Okay, you can check how much chromium 6 is there and how much is reduced to chromium 3. It's for all metal. Okay, it's like for selenium or cadmium. Whenever we are doing in the aqueous media, we always need to see, or arsenic, we always need to see that the reduction profile, and as well as it if it is reducing, it's reducing to what. Okay, so aqueous media you can simply go with that, but in the biomass you always have to go with XRD as well as XPS to know what kind of metal is there present in the biomass. Okay.
How many after running the experiment? On how many hour window we can perform this?
Bioabsorption is very fast. For bioabsorption, we generally go with 1 hour, or maybe sometimes maximum 2 hours. But at the very beginning, we take very fast, like after 15 minutes, after 30 minutes, because the bioabsorption is generally happens within I think 30 to 60 minutes. Okay, while for bio,
Joy, do you think bioabsorption is just like adsorption?
yes, is the similar surface precipitations. Okay, so that's why
We
similar.
have application somewhere like suppose we have tons of dead biomass and we are performing bioabsorption.
I haven't seen any real life applications. I have seen many lab scale study where people have column reactor and they have installed this column fill of biomass, dead biomass. Okay,
So
and they are starting the
they are filling the biomass.
yes. So that's why bioabsorption is like limitation is one, first you have to kill that thing. Second is the metal is not changing. Chromium is basically in bioabsorptions, chromium 6 will remain as a chromium 6. What if maybe after 2 hours, 2 years, it start leaching? It will remain as a chromium 6, isn't it? While if you reduce chromium 6 to chromium 3 and then immobilize, I mean chromium 3 can oxidize back to chromium 6 again, but that chance is much lesser to chromium 6 bioabsorptions process. Okay.
Another thing, sir, if we use the used tea bag as
Yes, so for instead of using dead biomass like biochar, okay, biochar is basically if you are like in wastewater treatment, we produce lots of solid waste, isn't it? Solid sludge. So what is the use of it? So many people are now trying to produce biochar from the solid waste, and that biochar is as Dr. Bipro or Dr. JP both told, that like very porous in structure. So they are very good absorbent material. Okay, so the biochar from the solid waste could be a application basically for the, because otherwise the solid waste has a very less use, whether it's a biochar or in abroad some people use in civil engineering as a biocement, the solid waste.
Reduce the carbon footprint.
reduce the carbon footprint basically. Okay, but so bioabsorption is another point. The limitation is it has a it immediately reach its equilibrium because it's the chemical precipitation. So the binding sites are fixed, it's not changing. Okay, because then we are doing with a dead biomass. So after a certain point it will reach its equilibrium or maximum stage, while for bioaccumulation it's never reached that saturation or equilibrium stage because the bacteria is alive, they are growing. Okay, bioabsorption is happening, they will bioaccumulate, so the surface become again free. So it's a continuous process, biomass is growing continuously. So that's why in bioaccumulation we can run the system for much longer time compared to the bioabsorptions. One is like a non-continuous and another one is continuous process. So this is biotransformation. I just gave couple of examples. The same thing, biotransformation or biodegradations. For organic contaminant is biotransformations or biodegradations. Okay, for metal we cannot tell biodegradations, isn't it? Because metal is non-degradable. So what happens in metal is the biotransformation or oxidation reduction, that's it, or precipitations. But the metal will remain there, isn't it? But for organic contaminants we call it biodegradations. So one organic contaminant will degrade into some other parts, isn't it? So this you can see that selenium can be reduced to selenium 0. Not only that, selenium can be reduced to selenide as well, just like sulfur. You have sulfur plus 6 plus 4, sulfur 0 and then sulfide. So it form metal sulfide and precipitate. Selenium has a very similar chemical properties like sulfur. Selenium 6, 4, selenium 0 and then selenium minus 2 or selenide. And selenide has a similar affiliation towards metal, or metal has a similar affiliations or interaction with the selenide. Okay, as soon as they find selenide is present, the metal will precipitate as a metal selenide. Okay, and in that way you can remove both metal selenide, metal as well as selenium. Okay, just like selenium, metal sulfide, why it is so famous in metal removal? Because you don't need to do anything, bacteria reducing the sulfur to sulfide and metal will immediately precipitate as a metal sulfide. So in this way you can remove both sulfur as well as metal. Same way, instead of sulfur you can use selenium as well. You can have mercury as well, the same examples. Mercury is reduced to elemental mercury which become less toxic. It can reduce to mercury minus 2 as well, it become less toxic. Arsenic is another example for oxidations. Chromium is another examples. But one problem in chromium is, or selenium, whatever reduced metal or oxidized metal you are producing is not like they are non-toxic. They are toxic but less toxic compared to their starting metal. Okay, so not just about reductions, we also need to find out how we can immobilize them. Okay, like for chromium there are so many researches going on, like if you heard of calcite precipitations. Okay, so with calcite or calcium carbonate, you can say microbial induced calcium carbonate precipitations, the chromium can get inside the same crystal and can precipitate as a calcite chromium 3 precipitations, and in that way you can immobilize the chromium. Okay, selenium is the selenium nanoparticles, you can immobilize with the biomass. Arsenic is the same thing, but in arsenic we have a very major challenge. Basically now so many people are trying to reduce the arsenic 3 into some other arsenic 0, which is very less phenomena, but some research has been published to immobilize the arsenic metabolites as well. Okay, so this one about, I will not repeat, the intercellular and extracellular sequestration or reductions basically. Biodegradation is for the organic contaminant. So I will just give you one example. Yes. So these are the, of course we don't worry about the biodegradable compound, whether different food waste or not, because they will degrade. The whatever microbe is present in the nature, they will degrade them today or tomorrow. Okay, so the organic contaminants is bit more easier to degrade. But one of the major problem in organic contaminant is, suppose you are starting with A, A organic contaminant. So if A is degrading, of course there will be some products, chemical reactions. Maybe A will degrade into B, C, D, different byproducts is there. So your A concentration in your media is reducing, while concentration of B, C, D will be increasing. Now in organic degradation, that is the basic or main challenge basically, to identify what are the byproducts. Okay, and the identification of them is very difficult. You cannot do with the basic instrument. For that you need always LCMS, okay, liquid chromatography with MS. That will gives you the chemical formula, and then you will design the what are the different kind of byproducts is present. But that byproduct could also be toxic in nature, isn't it? So you need to do basic test as well to find out whether the byproducts are toxic or not. You can, like, you treat the water, or you suppose you have a synthetic wastewater and some pesticides is there, you are doing biodegradations, or what Dr. Asutosh does is photocatalytic degradations. So one of the chemical degradation or biodegradation, one of the hurdle is we are still getting some byproducts which needs to be removed, because we don't know whether they are toxic or not. Or you can do it, suppose you are starting in a simple conical flux batch experiment. You have, let's just say you decide that 3 days of incubations is required. So you have one inlet or influent with the toxic wastewater. So after 3 days some degradation is happening, isn't it? And you can add microalgae or plant in both kind of water, and you can see their growth. In this way you can find out what is the toxicity, how much toxicity you have reduced, if it is not possible to find out the what are the degradation products. So just for your example, I don't know much about LCMS. Okay, it's like I did one experiment with tetracycline biodegradations. I did the LCMS in collaboration with IIT Guwahati professors, but I have the raw LCMS data and I need again his help to analyze those data.
Let me explain a bit quickly.
If you did with LCMS data, so
So actually, we take the sample and we analyze what kind of M by Z ratio is there, right? Then on that basis, we actually check the library. In that library, suppose M by Z ratio, suppose the first, yeah, belongs to this one, then we predict, okay, this is the compound. And then now once we have the name of all the compounds, then we kind of predict the pathway, like suppose carboxylation is happening or hydroxyl radical is getting added, then this compound will be formed. So that is how this
Is there any probable mechanism of getting the perfect product? This is actually in this case, first case there is probably 5 compounds. Is there any one probable mechanism that we can predict the perfect compound?
No. Actually, these all suppose 20 compounds are present. You have find through the LCMS, right? Then what is your parent compound and how these 20 compounds has come? That is how you are predicting with the help of what kind of reaction could have happened.
that tells you the mechanism, pathway basically.
Suppose you have benzene ring and you are getting benzene ring plus OH. Then probably you are predicting OH radical is coming and it is joining the benzene ring, then phenol is made, like that.
Okay, so here you can see, so this, I don't know if it is visible or not, like BBP1. So it's basically the biodegradation pathway 1, biodegradation pathway 2. So you can see for them they try to predict the ciprofloxacin biodegradation. But the main thing I want to see is show is they started with this structure ciprofloxacins, and you can see that they are getting different kind of 4 different major. There are many other byproducts, but these are the major 4 biodegradation product they were getting, and based on that they propose a hypothesis or a proposed mechanisms which mechanism could be followed. So this is a new paper from ciprofloxacin biodegradation. So they did basically bioelectrical system, which Dr. Bipro was teaching you, I think yesterday only, microbial fuel cells or microbial electrolysis. Okay, so based on, so they combine this catalytic degradations as well as biological degradations. One of the major issue when you are doing chemical degradations, your water is much clear, isn't it? In biodegradations, there are many other metabolites present. So we have to go with like sequential SPE or solid phase extractions, those kind of purification method before LCMS. And both, whether it's SPE or LCMS, they both are very expensive. Okay, you will hardly get, like if you have a very close collaboration, probably they will run 1 or 2 sample for you. Even if you are like very close to them, they won't. If you send 10 sample, they won't do it. Okay, and irrespective of how big he is or how much fund he has, it's a tedious process and expensive as well. Okay, so that is the basics of bioremediations. So I will focus little bit on phytoremediation as well, because we have studied from the beginning to till now. It's all about microbial bioremediations, whether it's pure culture or granule sludge in wastewater or microbial fuel cells. We always use mixed microbial communities because the mixed culture gives you the better results all the time. Okay, it's like the example I gave you yesterday is like fighting alone versus fighting with your team member. Okay, in phytoremediations, so instead of forget about all the bacteria, we use plant material. Basically, of course, the plant is living, so there is no bioabsorptions. Bioabsorptions can happen at a particular part, maybe root bioabsorptions could be there. So in phytoremediation, first thing, one of the major advantages, this is non-destructive process. Okay, like the garden, as the in situ bioremediation. If this garden, suppose some spillages happen with some pesticides or chemical toxic compound. So what will happen is that you can plant some resistant material or resistant plant biomass. They will grow over the time period, and in that durations they will uptake the toxic compound. Okay, so there what is most important thing is how you will choose the plant. In microbe is like we go for microbial growth, microbial resistance or sustainability, how much resistant it has. For the plant we have to go for the same thing. They are toxic, like how resistant they are. But not only that, in plant one of the major challenge is how fast they can grow. That is the limitation of phytoremediation. Plant can सर्टिफिकेट देना आ रहा है, बोलो EDC में पहुंचा दो. Okay, so for the plant, one of the major challenges how fast it can grow, because if you do some plantation a plant which may take like 10 years to grow to its place, so that means you need to wait for 10 years for to restrict the contamination over here. Okay, so anyway, so for plant we have various mechanisms which it follows, like phytoextractions. From the name itself you can understand volatilization, stabilization, degradations. It's like whatever we studied in bioremediations is the same thing is happening over here, but instead of microbes we are using different kind of plants. Okay, and after that, so suppose plant is extracting the metal from the soil, so the biomass or the metal will remain in the biomass, isn't it? And plant has a certain life, like maybe it will survive for 25 years. After that it will be dead, it will be killed, isn't it? So after 25 years what it will do, because the metal which is extracting, these are all accumulating inside its biomass. So if you just throw it, it will slowly start degrading. Decomposer is there. See, if it start degrading in the environment itself, so the metal which it accumulated over the time period, they will re-enter into the environment, right? So that's one of the major issue in phytoremediation. So either you can like collect the biomass.
Sir, can we use the biomass again after the plant is dead? The dead biomass can be used as a biomass.
You can use as a, you go for this experiment and you can see whether bioabsorption is happening or not, because the metals is inside, and when you are doing bioabsorption dead biomass, it will happen on the surface. Okay, though the surface properties of plant will be different. And you see like many like even like agricultural waste which is converted into same way biochar. There are many applications where agricultural waste, biochar or agricultural waste adsorbent has been used for metal adsorptions or different kind of experiments. Okay, there are many studies on it, but again the thing Dr. Ashutosh was asking, like the large scale application is still not there for bioabsorptions. Okay. So the one I was talking about, plant properties, which is very important. So these are the couple of points when you are selecting, because just like microbes, there are also thousands of different plant species available, isn't it? So how you will choose that what plant is best for you? Of course, it's not like you will choose one and you will start your experiment. You will choose 3-4 different species. Like microbes, you will choose 3-4 different microbes, bacteria to do the experiment. Same way for plant. So these are the properties of fast growth rate, high biomass production. So because the higher the biomass productions, the better is the accumulations. Okay, extensive fibrous root system. So what is that? So how plant will uptake it? They will uptake the metal or organic contaminant through its root system, isn't it? So the more extensive or the more distributed it is, the more area it will cover, isn't it? It's not like the root is only in this region and your contamination is there. Root will extract the metal from that far distance. So plant will do it from this rhizospheric area only. So the more extensive root system you have, fibrous root system as well as extensive one, the better it is. Easy to cultivate and harvest, isn't it? It's not like a plant which is very like soft to the nature. Sudden change in temperature it will be dead, sudden rain it will be dead. You don't need those kind of plant. You need a robust plant which doesn't need too much care, isn't it? Because if it is a normal toxic, it is okay for you. But suppose imagine you are working with a radioactive waste. You don't want to go there each and every day to monitor, isn't it? Just from outside you will check, and maybe once a month you will do some cultivations. Otherwise plants should be grow by themselves without much care. Okay, that's why it is very important for us, so that we don't get easily contaminated. Okay, highly tolerant to contaminants. Why? It is very important, because if plant is not tolerant, plant is accumulating the metals. As soon as if its toxic limit is very low, it will be dead maybe after 6 months. Okay, because it is accumulating the metal. So the tolerance level has to be very high for it. Okay, that's why you have like hyper accumulator plant as well. You have normal plant, you have tolerant plant or accumulator plant, and then you have hyper accumulator plant. Hyper accumulator plant is also, it's a very important part of research in phytoremediations to find out, and if you have it, you can get a very good paper.
Like cherries accumulate 1000 mg.
Yes,
Of arsenic.
arsenic. So it generally from like 100 to 1000 times higher than normal accumulator plant. Okay, so how you can, so for that also you have different kind of experiment you can study, like bioconcentration factor. So if you add, suppose you are doing pot study, okay, in your soil you add arsenic and you are growing some plant. So of course after maybe let's just say after 6 months, arsenic will be accumulated. You know how much arsenic you have added in the soil. So after 6 months what you will do is, you will remove the soil, you will separate the plant, right? So then you will analyze how much arsenic is present in the soil, how much arsenic is present in the plant. That ratio will give you bioconcentration factor. Then you can also study like translocation factor. What is that? Because when plant is uptaking the arsenic or any metal, so it will be uptaken by the root, isn't it? Translucent means the arsenic will be transferred from the root to the shoot, right? So you have separated the plant. Now you can separate the plant into root, divide. So root is in one side and the upper portions, and you can measure how much arsenic is present in the root, how much arsenic is present in the upper portion of the plant. That ratio will give you the translocation factor. So this kind of various parameters basically tells you about whether the plant is the accumulator plant or hyperaccumulator plant. Okay. So these are a couple of limitations for phytoremediations. Of course, it is a very time consuming process, because bacteria or microbes basically they have a very faster growth rate, isn't it? But plant, whatever you choose, it cannot be done in one day, right? It cannot be done in 2 days. So that is one of the major limitations of plant. Then also it's not like it can work against all kind of metal or all kind of organic contaminants. That is one limitation is there, very restricted, or the diversity is much less what kind of metal or what kind of toxic compound we can use. So this is the mechanisms of phytoremediations. So you can see that two mechanisms happens below the surface, below the ground. Okay, so one is phytosequestrations, one is phytostabilizations. Okay, then you have phytoextractions. From the name itself you can understand it extract the metal. So same thing like bioaccumulations for bioabsorptions. After bioaccumulations, bacteria do biodegradations or biotransformations for their detoxification mechanism. Same thing for plant. Plant is extracting metal or organic compound. It is toxic to them. So plant will apply some detoxification mechanism, and that is nothing but maybe phytovolatilizations or phytodegradations. Okay. In phytovolatilizations, you can see that of course if some reductions is happening like mercury. So suppose plant is uptaking the mercury +2, then extraction is happening and then degradations or volatilization is happening. So mercury 2+, when it reduced to 2-, it volatilized, it leave into the atmosphere. Okay, so that is like completely you are reducing the mercury concentrations in soil. Similarly, there are no mercury present in the biomass, so you can distract the plant as well, there is no issue with that, and mercury is releasing into the atmosphere. And that's the one like critical assessment needs to be done, because there are so many people who like ask you like, if the mercury 2-, is it not toxic, is it not polluting the air or not? Okay, so those kind of debate is still there, but again the thing is that -2 dimethyl mercuric oxide are much less toxic than the mercury 2+. Okay, so these are the whole mechanisms basically how plant do the phytoremediations. So in phytosequestrations you can see there are 3 different mechanism is there. Okay, one is the phytochemical complexation. So root release different chemicals. So it's like a chemical precipitation of metals. Okay, as soon as metal comes in contact with those chemicals, it form a complexation just like simple bioabsorptions, and the metal complexation will remain on to the rhizospheric zone. So basically you are not in the phytosequestration, you are not removing the metal or contaminants, they will remain in the soil itself, but you are restricting them, you are immobilizing them. Like this garden got contaminated, if you don't do anything over the time period, because the metal is mobile, so the groundwater, surface water, through that the contamination will spread it slowly all over the area. So with this phytosequestration or phytostabilizations, we are not removing the contamination from this garden, but will restrict their movement. This garden is contaminated, let it be. Okay, so that is the main difference with other method. Okay, then another mechanism is transport protein inhibitors. So it's not like that plant have all 3 mechanism at one plant. It can have maybe only one of those, or maybe all 3, maybe only 2, depending on the plant species. So these are the different mechanisms they follow. Okay, but these are the main 3 mechanisms for the phytosequestrations. It's like transport protein inhibitor on the root membrane. So the plant is uptaking the metal via root. So there are some protein inhibitors there which restrict the transfer from the soil inside the root. They will trap the metal on the root surface itself. Then you have the vacuolar storage. So if it still escape that part, or maybe some plant don't have all those two mechanisms, okay, they have only this vacuole. So if it is come inside the root, so they will restrict their movement from root to the upper part. How? They will trap them inside their vacuolar storage, and so that means if it is in the root, again is the same thing, it is within the soil itself. Okay. Same for phytostabilizations, it's the very similar mechanism. Only thing is it doesn't require all those transport protein inhibitor or vacuolar storage. It completely stabilizes the metal or contaminant on the soil itself, on the rhizospheric zone. The metal or contaminant doesn't go inside the root at all. Okay. So the thing is, as I here highlighted it, one of the major limitation is contaminant remain in the soil. So suppose after 5 years your plant is dead, you are taking out the plant, so the contamination remain in the soil, isn't it, and they can start leaching again. Okay, so that's the one major issue. But another important not issue but advantage is, suppose you are going for extractions. Okay, in this mechanism, suppose you are going for extractions, suppose this plant is edible one. So what will happen? There are lots of wild animal is roaming. So if they eat this plant, suppose it is extracting arsenic or extracting chromium, cadmium, whatever it is, it's present in the plant upper body, isn't it? If the animals are eating it, that means now it is entering into the food chain, isn't it? So you are doing more harm than the previous one. Previously at least it was present in the soil only, but now it is entering into the food chain. So when you are following only these two mechanisms, phytostabilization or phytosequestrations, you don't need what kind of plant you are using, because it is in the below ground. But as soon as you are following these three mechanisms, degradations or volatilization or extractions, you cannot use any edible plant for phytoremediations. That is one of the major thing you have to follow. Okay, in these three mechanisms you cannot ever use a edible plant. Okay, then it will be a complete disasters. Okay, so every time I see here some examples is given, I can't remember the local name. The botanist, I think he can tell you some local name of these plants. Okay, the phyto extractions, so these are the properties: fast growth, high biomass, high tolerance, and you can see that it must be non-edible to avoid food chain contaminations. Again some examples. So it's not like that phytoremediations hasn't been used. Phytoremediations has many large scale real world applications people have used to contain the contaminations. Not only that, even in solid waste also, so many people, like simple constructed wetland, you have probably heard of constructed wetland. So in the constructed wetland, in the upper part is plantations. So it's not only doing the phytoremediation, but it also have a aesthetic value, isn't it? In the solid waste, there are many examples in India also, where municipality dump solid waste maybe for 10 years. So when it was full, they then turn it into a garden. Okay, so it's not just for the garden purpose, it's not just aesthetic one कि चलो तुमने garden बना दिया. It's also that plant is doing phytoremediations below the garden as well. They are converting the toxic compounds into non-toxic ones. Okay, so plant phytoremediation has many large scale. You can just search and you will find out. So this is one research which you can have a direct real world application immediately, apart from that bioreactor which is already been seen. You have seen that, I think, into the industry visit we have been. So there is the hydroponic phytoremediation was going on. So in the hydroponic system we generally call them rhizofiltration. Okay, Why is the same? Same, it's the part of phytoremediation only, just the mechanism is known as rhizofiltration because it's the water. Here whatever mechanism we discuss is from the soil, from the solid, but from the water when you remove something we call it filtrations, isn't it? Water or any liquid media, and the filtration is happening by using roots, so that's why it's known as rhizofiltrations. Okay, so phytoextractions, then these are the some mechanisms of phytodegradation. So after extractions, the metals or organic contaminants are getting accumulated in the plant. So how to survive? They have these different degradations mechanism. You can see couple of enzymes which are present in plant which degrade various toxic compound. You can see aliphatic, aromatic compounds, oxygenase, and so it converted into from A to B plus C or D, or for metal again it will reduce to something, chromium, it will reduce to chromium 3. Okay. Phytovolatilizations I already discussed. So one example so I gave you is like typha. You can see that it so selenium contaminated soil, if you have, then it will convert the selenium into dimethyl selenide, and then dimethyl selenide is very volatile in nature and it will escape into the atmosphere. Similarly, you can see the mercury. So these are like, you have not just in research, you have a real application as well in large scale applications. Industry have already used it, okay, for these things. But the main problem is the one I was talking about, that what about this dimethyl selenide or mercuric oxide or arsenic, whether they are toxic or not. If they are toxic, how much toxic it is. If they are less toxic, how much less toxic it is. Okay, how much air pollution it is causing. Okay, so this kind of research or further risk assessment is required for phytovolatilizations. Okay, so I will stop it here. The basics of bioremediation and phytoremediation, just for those who are coming from outside. These are the different PhD or project is going on in my lab currently. So as you can see, it's mostly with the wastewater treatment, but nowadays we are not only working on wastewater treatment but also combining wastewater treatment with circular economy or sustainable development, isn't it? So this is one project of SBR sequencing batch reactor which working with pharmaceutical wastewater treatment. Then another is microalgae. So microalgae biomass has a very large use, whether you can go for the bio oil or you can go for biochar or you can go for biofertilizers. Okay, so one research is like selenium and fatty acid enriched microalgae production from wastewater, and we can use it for in agriculture as well as aquaculture as well. Then another PhD topic is going on hexavalent chromium removal from industrial wastewater. So this particular project is for using for this calcite precipitation, MICP, microbial induced calcite precipitations with chromium. Okay, so we are not only reducing the chromium 6 to chromium 3, but we are also immobilizing the chromium 3 with the help of calcite crystals. Okay, then so another one with the so microalgae 2 project is going on. Basically one is for wastewater treatment and for aquaculture. So aquaculture means feed source for different fish species. Okay, and this one is for agriculture. So we'll see how it can influence the biofertilizer as a biofertilizer. Then the biochar, which we already discussed. So what is going on over what in wastewater treatment? There are different method like SBR or biofilm reactor or simple activated sludge reactor, isn't it? So different reactor will produce different kind of sludge, and different kind of sludge has a different surface property. So when you produce them into biochar, the biochar can have a different surface properties which lead into your different removal efficiency, and then you can modify them. Some chemical modification could be there. So we are studying a different, so we collected different sludge from different wastewater treatment. We are producing biochar and then studying simple bioabsorptions for pharmaceutical or pesticides removal, and as well as also in agriculture, because in soil we have various contaminations, isn't it? So the biochar can influence the microbial communities, can immobilize different contamination as well, isn't it? And it can, in terms, it can, although we don't know how it will have a positive impact or negative impact, but hypothesis is if we add biochar, this kind of biochar, because they have also lots of nutrient as well, this kind of biochar should have a positive impact on agriculture. Okay, so that's another one. And this is completely related to sustainable agriculture. So we have isolated selenium resistant PGP. Okay, so from where I isolated, so the Punjab has the one of the most contaminated selenium soil in all over the world. Okay, so that was the best area. Arsenic, West Bengal is very famous for arsenic contaminations. So it's majorly the throughout the Gangetic area, okay, all area, and then you can have towards Bihar as well, the border, okay. Same in
Arsenic is the problem in those areas.
Purulia. I mean, not only Purulia, all over the West Bengal, basically arsenic is a huge, huge issue, and all the arsenic source is nothing but the anthropogenic. Okay, it's not like people are responsible, people did something and the arsenic contamination is happening. Okay. Oh yes, yes. So in UP also there are 40 or 45 different locations are people have recently surveyed it. Although UP is very weak, so if you compare with West Bengal, that's why it's not like that. In other states there are no arsenic pollutions.
Even Bhatinda in Punjab is highly contaminated with arsenic.
Yes.
I will tell you in Bhatinda not even arsenic. In groundwater you have.
They have uranium is also in Punjab. So in Punjab pollution is, I think it's mostly happening because of too much water extractions from the
Excessive usage of pesticides
because of that. Okay, so that's why here we try to isolate this different metal resistant PGP, and we are trying to form microbial communities. So one is with maybe selenium resistant, one is arsenic resistant, one is normal PGP, different growth, and we're of course we cannot aim at all metal at a time. So yes, so cadmium, lead, all is there basically. Selenium I choose because it was my like old interest. For last 10 years I'm working on selenium, and arsenic is I am from Bengal, so I know about the arsenic contamination situation in Bengal. So in this way basically, otherwise you can choose whatever metal is your interest, but you cannot choose for one PhD you cannot choose all different metals. Okay, maybe for other PhD you can go for other metals. So these are the different projects right now going on in my lab. So that's it. Thank you. So if you have any questions. You will show something or just?
I can just I can actually use this slide.
Okay. So now Dr. Bipro will take I think another half an hour just to review and show some slides. So these are of course not part of the exams which you will have later on. So this is, take it as a normal discussion, so you can ask, and of course after that you will be leaving, so utilize it to the fullest basically. You haven't done that?